Okay, we're going on now with this book right here. It's called Prevent and Reverse Diabetes by me, Pete Rogers. We're on part 6. I actually skipped a chapter to chapter 5. The reason I skipped it was it's a real technical chapter with no pictures, because I kind of write my books for several different audiences. Number one, I want it for the patient to help them to get better. But number two, I also write it for doctors, for skeptical people, and I'll have some chapters like the last one. It's real technical with a bunch of footnotes. This is a chapter I think everybody will benefit from and enjoy if you're interested in diabetes. Okay, I actually just called this entire chapter Pictures of Diabetes. So this drawing here shows a staging system of diabetes. It's based on the ideas like of Gerald Shulman, 2018 Banting Award winner, as best diabetes researcher in the world. He's got a great YouTube video, by the way. You can find it on YouTube. Just type in Banting 2018 lecture Gerald Shulman. Okay, but the point of this is it's also been called the ectopic fat theory. So the good way to think of diabetes, first of all, get it out of your head that it's a sugar disease. That's only what stupid people say. Diabetes is well known to be a fat disease. It is a disease of fat metabolism that causes increased blood glucose and then all these secondary complications. The problem begins with excessive fat in the skeletal muscle. When you have excessive fat in the skeletal muscle, shown right here, that's been confirmed with nuclear magnetic resonance spectroscopy. It's been known for a long time. Michael Brownlee knew about it. All right, he was the 2004 Banting Award winner. That causes insulin resistance because, in a sense, it's overnutrition. Too much fat gets into the skeletal muscle. They call it intramyocellular lipid. And then the mitochondria sort of become overwhelmed by it. They send a signal to the cytoplasm to shut down the uptake of glucose into the cell. So the glucose type 4 transporters don't go to the plasma membrane. Over time, you start accumulating more fat in the liver. That's called fatty liver. Fatty liver is prediabetes or diabetes of the liver. Then you start accumulating fat in the pancreas. And then that destroys the pancreas beta cells, which eventually completely lose their ability to produce insulin, or a significant amount of insulin. Then the patient's insulin dependent for life. So that's why if you have type 2 diabetes, you want to reverse it as fast as possible so you don't go on to lose your pancreas. All right, the typical stupid person says, I'm taking my pills, my sugars are okay, everything's under control. No, you're still losing your pancreas, you still have insulin resistance, you're still developing atherosclerosis throughout your body. The smart move is go low fat, low sodium, vegan with no oil, and reverse it if you can, if you still got enough insulin function in your pancreas. And even if you're a full-blown type 1, type 1.5, you still want to optimize your diet and your health habits so that you minimize your insulin resistance, you minimize your insulin medication requirements. There's a lot of benefits to doing that. I'm just going to go right to the next picture because I'm kind of giving you all the explanations in between the pictures. Here is a mitochondria, and the outer black line is the outer mitochondrial membrane. The red arrow points to the intramembranous space in between the outer membrane and the inner membrane. The inner membrane is this green line. That's the inner mitochondrial membrane, it's abbreviated IMM. The yellow stuff in the center is a mitochondrial matrix. So inside the mitochondrial matrix, that's where Krebs cycle runs. Krebs cycle is also called tricarboxylic acid cycle, also called the citric acid cycle. This is where about 90% of the ATP in your body is produced. So it's a really important little organelle. It's fascinating. I've given several other lectures on it, including a couple recently. My series of lectures, Mitochondrial Insights, are a good one to watch. I've only done 2 parts to it yet, but there's other parts I'm planning to make soon. Plus, I've given it other lectures about mitochondria inhibitors and whatnot. But for our purposes right now, what you're going to need to know is that insulin resistance is primarily due to fat accumulation in the skeletal muscle cells, overwhelming the mitochondria. Here's a quick look at the difference between fat and a glucose. So here's a glucose opened up called a Fisher projection. I remember Fisher projection because it's like you're tossing a fishing pole out straight, as opposed to when it's around like a cyclic ring, like a Hayworth projection. Okay, I remember that as Rita Hayworth, the female actress, like a breast looks like a round thing. Okay, that's how I remember it. Cyclic versus open chain glucose configuration. Here's a fatty acid, a saturated fat, palmitate acid, C16. That is the most common saturated fat in the human body. Saturated means there's no double bonds. A fatty acid is basically a carboxylic acid with a long alkyl chain, just carbons and hydrogens behind it. So it's very non-polar. You can see carbohydrate is a hydrated carbon. There's a water on each carbon, H2O on each carbon. Okay, versus fat, it's very dry. There's no water on there. And because of that, you can store it dry. You don't need to add water. The liver can only hold enough glucose to support you for about a day, day and a half in terms of glycogen storage. But your fat can keep you alive for 60 days, okay, in starvation. And because it's much more reduced, you generate energy by oxidizing it. So it doesn't have any oxygens on there. You can store it dry so it's lighter weight to store more energy. In the liver, you have to store glycogen with water attached to it, a couple molecules of water. So that's why the liver, despite how giant it is, it really doesn't store that much glycogen. That's the main job of the liver, to maintain blood glucose level during fasting for the brain. Okay, here's a quick look at glycolysis. Glycolysis is the first biochemical cycle involved in the processing of glucose for energy. You basically take a molecule when you want to process it for energy and you chop it up into pieces and burn the individual pieces. It's not like if you start a fire, you burn everything at once, because you wouldn't be able to extract much energy from that. You would just get heat. But by chopping off CO2s one at a time, you can extract a lot of energy. The energy gets converted into electrons. Those electrons are sent through the inner mitochondrial membrane. We'll come to that in a moment. Here's what happens with glucose. So it gets brought into a cell and it gets phosphorylated. Okay, the enzyme is hexokinase, or you can call it glucokinase. Then it becomes glucose 6-phosphate. The next reaction, it isomerizes to like fructose. I call it a fructone because it's a ketone. The key point is it's a more symmetric molecule to get ready for the second half of glycolysis, where it's split in half to two nearly symmetric parts, okay, and then they become symmetric, okay. That's how glycolysis runs. All you need to know is glycolysis is run without oxygen. You need to know that. It's anaerobic. And to metabolize glucose solely by anaerobic glycolysis, that's called fermentation. Okay, you need to know that too. Whereas when you burn it in the mitochondria, you need oxygen, and that's called oxidative phosphorylation, or you call it respiration. So those are two ways to discuss glucose metabolism. Fermentation means anaerobic, respiration means with oxygen. Okay, you need to know that. That's going to come up a lot. Okay, this just shows some more reactions down the pathway. And the one thing you need to know is that sort of the heart of glycolysis is this molecule right here, 3-phosphoglyceraldehyde. It's an aldehyde, 3-phosphate glyceraldehyde. That is going to come up a lot because the complications of glucose, the most important ones, derive from an accumulation of this molecule when this enzyme is inhibited. You make 2 ATP per each molecule of glucose. That's also worth knowing because you make 18 when you can use oxygen in the mitochondria. So you get a lot more energy out of the mitochondria. Okay, then I was showing you this one reaction in more detail here, glyceraldehyde 3-phosphate, and it's metabolized by this enzyme, glyceraldehyde, that's the GA, 3-phosphate dehydrogenase. This is very important because this is what's the main thing that's getting inhibited in glycolysis when you have diabetes with insulin resistance. There's other problems happening simultaneously, but this is the big one. Okay, here's just some other things that can inhibit this enzyme. So PARP is the polyadenylated repair enzyme. That's the one that blocks it with typical insulin resistance. But with drinking excessive amounts of alcohol, you can also get inhibition of this enzyme. All right, arsenic can also inhibit this enzyme. So that's important to know. All right. We talked about how you minimize arsenic toxicity on foods by rinsing them and avoiding ones that are high in arsenic. Okay, then in the end of glycolysis, you end up in pyruvate. If there's no oxygen available, the pyruvate's going to get converted to lactate, or lactic acid plus a proton, lactate plus a proton, lactic acid. Okay, that's going to get pumped out into the extracellular matrix. That's why anaerobic metabolism acidifies the extracellular matrix. Okay, but pyruvate is the key end product of glycolysis. So in anaerobic metabolism, it goes to lactate. In other situations, it can go to different things. Okay, under normal oxygenated conditions, it goes into the mitochondria as acetyl-CoA, and then you can run Krebs cycle. But arsenic will also block that. So arsenic can do a significant amount, but arsenic is usually exaggerated as a toxin. Dr. McDougall said he never saw a single case in his whole life, and he lived in California where there's a lot of rice-eating Asians, okay? I've eaten rice plenty of times. I've never had a problem from it. So it's worth knowing about arsenic. There's actually more arsenic contamination of other foods, but everybody talks about rice because they want to try to demonize a good food rather than talk about the foods they don't want you to know about. It's in a lot of other things too. Okay, okay, next thing here. Glucose comes into the cell, hexokinase phosphorylates it, that traps it in the cell, putting a big charge on it so it can't exit out. Glycolysis converts it to pyruvate, then it gets made into acetyl-CoA, it's in the mitochondrial matrix, goes through Krebs cycle, and then the electron carriers are sent to the inner mitochondrial membrane, which passes those electrons along like a fireman's bucket brigade. And again, now if you are worried about diabetes, all you need to know is the things that cause insulin resistance and how to avoid them. I'm going through all this stuff just to give the people who care a little bit more sophisticated understanding of diabetes, because it helps you to understand some of the less common types of insulin resistance. So here is normal respiration. So what that means is the electrons are passed through these carriers, and they take the energy acquired by passing the electron downhill, sort of like a snowball rolling downhill. And it goes to a progressively more aggressive grabber of electrons. The ultimate acceptor of electrons is oxygen. It's the second most electronegative element in the periodic table of elements, meaning it wants to grab electrons very forcefully. And so as you pass these electrons down, it's like passing electrons down a wire, oxygen grabs them, and then 2 hydrogens are added to it and it becomes water. Okay, but in the meantime, these protons have been pumped out into the intermembranous space between the outer and the inner mitochondrial membranes, and they accumulate there, generating proton pressure, okay, because there's a big gradient, and it's highly electrical, and it's a concentration gradient as well. The electrical gradient across this membrane of the inner mitochondrial membrane is about 160 millivolts. That's a giant gradient, the most powerful gradient in the whole human body. And what the mitochondria is essentially a nuclear power plant. You split the atom of hydrogen into its proton and its electron. The proton is pumped into intermembranous space. The electrons are passed down a wire, down the electron carriers, like a fireman bucket brigade, until they reach the ultimate acceptor of oxygen, and oxygen gets converted into H2O, and then a proton is pumped back through ATP synthase, and it spins like a mill wheel, like a rotating revolving door in a big hotel, and that energy of that spinning is coupled to pushing a phosphate onto ADP, adenosine diphosphate, as in 2 phosphates, to become adenosine triphosphate, as in 3 phosphates, and this is the energy currency of the cell, like a $20 bill. Okay, that's how it works. That's life on Earth right there. I know that might have seemed mundane, it might have seemed complicated if you haven't heard it before, but I'm telling you, this is like one of the most important things you could ever know about life on Earth. The guy who figured this out was Peter Mitchell, an Englishman in England in the 1960s. He was a work of genius, okay, to figure this out. And then they realized, gee, all life on Earth depends on this. The bacteria, the plants, everything depends on these proton gradients. Okay, this is just another slide showing pretty much the same thing, but I've got the gradient written in there, negative 60, 160 millivolts, okay, for this little nuclear power plant that splits the atom, okay, and then generates electricity. It's quite amazing. If you got the smartest chemists, physicists, and all of them on Earth, they couldn't make this. Only God could make this. The more you learn about this stuff, the more you believe in God. It's beyond amazing. It's so incredible. And it's also very beautiful and elegant. Okay, here's what I meant by the periodic table of elements. The most reactive molecule in the entire periodic table of elements is fluoride. It's so aggressive that it's in the vertical column for the halogens, which basically kill stuff. Okay, all of these things are used to sterilize things and kill things. Okay, oxygen is next to fluoride, is the next most aggressive grabber of electrons. The number here written at the bottom is the electronegativity. So oxygen has a very high electronegativity, desire to grab electrons. Okay, then here's just one more diagram of the electron transport chain. And what it basically shows is, the idea of a nuclear power plant. The hydrogen atom has been split with the H plus component from the nucleus, and its protons are pumped into intermembranous space, IMS. The electrons, that are outer part of the molecule that orbits the central nucleus, they're passed down through these carriers. And then the gradient is harvested by letting one proton come back in, and the energy that generates spins this like a mill wheel, and that's coupled to adding a phosphate to ADP to make ATP. Okay, so I'm going to leave it at that for today, and then when I come back for the next one, I'll go through more of this sort of thing and start explaining in more subtlety the nuance and details of the mitochondria, how it functions and how this relates to diabetes.